Yeah, now we are on.
Let me just like switch off the sound at YouTube because normally it's on. Yes, it was. So good afternoon, everyone. For us, is a real pleasure to have Professor Subhash Minocha for our talk today. I will not introduce Professor Minocha because that's the role of Leandro introducing our speakers. So I just would like to thank you, everybody who's here, the students that join us at Google Meet, and people outside on YouTube joining for this very nice talk, I'm sure. I would like to reinforce the information that the students here can let your questions either in English or in Portuguese. Feel free to choose on the chat here at Google Meet, and also there in YouTube. You can also ask in English or in Portuguese that we can translate it to Professor Minocha. So, Professor Minocha, in the name of the whole team, I would like to thank you for your talk today, to accept our invitation, and I will give the word to Leandro for your introduction. Thank you very much.
Thank you, Igor. Briefly, Professor Minocha received its PhD from the University of Washington, Seattle in 1974. After that, Professor joined the University of New Hampshire, where he has been a professor of plant biology and genetics. During his tenure at the UNH, he has been involved in teaching a variety of courses in plant physiology, plant developmental biology, plant biotechnology, biotechnology society, cell culture, and plant growth regulators. Also, he has researched on a broad range of projects like cytodifferentiation of somatic embryogenes in several species using techniques of biochemistry, DNA manipulation, genomic, transcriptomic, and metabolomic to understand the metabolic regulation. Also has established national and international collaborations in research on 5 continents. Wow. And serving as a department chair in Botany and plant physiology, pathology, and program director. He has mentored more than 35 graduate students and more than 250 undergraduate students for research. Professor, it's a honor for us to your presence in your course. Also, I would like to comment that here in Brazil, today is the National Day of Science and Day of Scientific Research, so it's a good day for us today. The class is yours, the students yours. Feel free to speaking.
Well, thank you very much, Igor and Leandro, for inviting me. It is a wonderful feeling to be invited on such an international kind of a activity that you have organized. I'm very impressed with the number of people who have shown up in your program, and I'm glad to be here. So by way of just a brief comment, I would like to just give you an idea of what I'm going to talk about. Of course, the target group of chemicals is called polyamines. And we will give you a good summary of what we have been doing over the last several years and why we are doing it and where do we stand. And hopefully there'll be some discussion on where the field is going and what we can do. So let me, I have to start presenting, right?
Not yet.
Can you see my screen now?
Yes.
Okay, so the topic is basically getting high on putrescine. Now, we all know in English the word getting high means when you get drunk, what happens to you? Of course, plants don't get drunk with putrescine, but the idea is the same, that if you have a high concentration of something, how do you react to it? And our goal is to make the plants respond to changes in polyamines, particularly in putrescine. So the title is Metabolic Engineering of the Polyamine Pathway. I should point up front that a lot of this work has been done in collaboration with Dr. Rakesh Minocha, who works with the USDA Forest Service. And she and our lab have been collaborating for quite some time with a lot of students getting advice collaboratively from both of us. Just to give you an idea of the background, polyamines. What are polyamines? Polyamines are low molecular weight aliphatic amines, which are present in all living organisms, including viruses and including the coronaviruses. They are integral for survival. Putrescine, spermidine, and spermine are the dominant polyamines in living organisms. There are a number of other polyamines, but most of them are minor in quantities and also variable in different organisms. Polyamines play active roles in DNA and protein synthesis, membrane and nucleic acid stabilization, free radical scavenging, nitrogen metabolism, tolerance to environmental stress. That covers a lot of things that the cells use the polyamines for, and that's what makes them very interesting, and that's what makes them very confusing. Because if something has a specific role, you can target what is going on with that particular chemical and its interaction in the cell. When something does almost everything, it's like, what does sugar do in our cells? It does so many things that it's hard to say what really is or are the functions of polyamines. Almost everything depends on them. And since polyamines are required for cell division, and their cellular content is very high in all types of cancer cells, this is well known for the last couple of decades, inhibition of their biosynthesis actually is a target of cancer chemotherapy. There have been many chemicals that have been used, tested. Many of them do not work for humans because we cannot control their diet. Our diet, especially plant sources as well as animal sources, are very high in polyamines, and I'll make a comment about this point later on. Polyamines are also a marketing theme for a lot of nutraceutical companies. And here's from one company called Oriza.co.jp. Just got that picture from there. Beauty enhancement effects of polyamines, activity to prevent atherosclerosis, promote fat metabolism, health revitalizing, promote longevity, enhance cell proliferation, you name it, everything. And they sell polyamines in the form of pills or powder that you can get to feel good, but being careful once again that too much polyamines can be actually more connected to certain diseases as well. So here are some supplements that they sell to at least add the commercial aspect of the theme. Natural ingredients for healthy hair and nail treatment and anti-aging. Polyamine P, water-soluble powder, wheat polyamines, you name it, everything in the market is up for sale. But anyway, from plant's point of view, why are we interested in its metabolism? First of all, we said its diverse role in development and stress response in all organisms. Polyamines have a specific relationship to plant stress responses and nitrogen and carbon assimilation. And I'm going to emphasize this a bit more, because if we can increase nitrogen metabolism, nitrogen assimilation through changes in polyamines, through metabolic engineering, we expect we can get changes in carbon, more uptake of carbon. That means we can get more biomass. And that's where our work with poplar comes in, because poplar is a tree species that is grown over millions and millions of hectares all over the world as a source of biomass. So that'll become towards the end of the talk. Manipulation of polyamine metabolism in plants could help us or hurt us if we don't do it the right way, because it's easy to increase polyamine levels through genetic engineering. And as you saw in the previous slide, polyamines are quite important or play a major role in the development and the growth of cancer cells. So if you are a person going through chemotherapy and you are eating food that is rich in polyamines, you are going to be having much more effect on cancer and less effect of the chemotherapy that you are taking. So one of the targets, as we talk about in our lab, is to make food that is specifically low in polyamines in the parts that we eat, and it could even be used as a special diet during chemotherapy for cancer patients. And of course, there's the evolutionary aspect of polyamines, that they have been important in all living things. So here's the pathway that many of you probably are familiar with. The enzymes and genes that are involved in the core polyamine pathway. The major reaction that is present almost universally in all living organisms is the conversion of ornithine into putrescine by the enzyme called ornithine decarboxylase. And this is what I will talk about today, manipulating this enzyme by transgenic means. Putrescine then later is converted to spermidine by the enzyme called spermidine synthase, and spermidine is converted to spermine by the enzyme spermine synthase. And these two steps require decarboxylation of SAM to produce decarboxylated SAM, and that is done with the SAM-DC, or full name is S-adenosylmethionine decarboxylase. Plants and some fungi and some bacteria as well have an alternate pathway to make polyamines, and that is directly from arginine by the enzyme, starting with arginine decarboxylase, followed by a couple of more steps to give putrescine, and that's how you see it. This pathway becomes convergent, and we have three polyamines. The structures are given here. Not very important for today's talk, but just to give you an idea of what they look like. What are the special features? Once again, it's a repetition of what I said to make it more precise target. The two active pathways for putrescine biosynthesis, balance of organic nitrogen and carbon in the cell is very important, and polyamines are involved in at least contributing to it, if not completely controlling it. Polyamines help us in ammonia toxicity removal because they can use ammonia directly, and ammonia does not cause toxicity. And more importantly, in plants, there's a special need for hydrogen peroxide production in the cell wall. And hydrogen peroxide, one of the major sources of hydrogen peroxide is the degradation or catabolism of polyamines. So polyamines becoming that way quite important too. And then in plants, there's a very active putrescine to GABA pathway. GABA, gamma aminobutyric acid, typically is made directly from glutamate in animals as well as in plants, but the first product of putrescine degradation is also GABA. So there's a fair amount of work done on that aspect of polyamine pathway as well. To point out, after talking so much, polyamine pathway is not the center of the universe. It is on the side of the major pathways. So this one shows you right here that the main source of polyamines is glutamate, which is coming from nitrogen assimilation. And this glutamate is converted to ornithine through multiple steps, which will be shown in the next slide. Ornithine is converted to arginine. Arginine can be converted back to ornithine. Proline is another amino acid, very important. Here's GABA. So you can see how the pathways are interrelated. And our focus is what happens if you just add one enzyme, that is, take away ornithine from this whole reaction mix and increase putrescine. What happens to the rest of it? And you will see some results on that as well. These are some names of specific enzyme and specific reactions. If you're interested in the biochemistry, I'm not going to go through that. The main thing to point out is there are a number of enzymes, 5 to 6, that make ornithine from glutamate. This pathway is reversible to some extent, and ornithine then goes to putrescine and arginine and everything else. Here are some assumptions that we made when we started working with polyamines almost about 30 years ago. The assumptions are, since polyamines promote transcription and translation, very well known for many, many years, their increased biosynthesis should promote growth, and that is something that is seen in most plants and animals and microbes. Since polyamines stabilize cell membranes and help scavenge free radicals, their increased biosynthesis should help us in stress tolerance, should help the plant in stress tolerance, and that is a well-known effect of polyamines. Since nitrogen metabolism is causatively related to carbon metabolism, photosynthesis, that is, increased polyamine biosynthesis could increase carbon assimilation, and that's what we are working on more recently. And since polyamine biosynthesis is a target of chemotherapy in human cancers, I mentioned earlier, reduction in the polyamine supply in our food should be beneficial, and inhibition of their biosynthesis in parts of the plants used for food production will be desirable for cancer plants. So that's what drove us to spend more than 3 decades on the polyamine research, still continuing, and with some modifications. We have been very happy with the assumptions that are coming out to be correct in many cases. So what's our theme from a scientific perspective? It is taken from a very well-known saying from one of the US presidents, but modified that. Ask not what polyamines can do in a cell. Ask what disturbing their metabolism can do to a cell. So that is the theme with which we worked. That yes, we know polyamines do a lot of things. Now the question is, if you mess up polyamine metabolism through genetic engineering, what effects it could have. Objectives, specific objectives of our study are to study the effect of altering polyamine metabolism through genetic engineering on the biochemistry, particularly of polyamines and amino acids of plants, to study the effects of altering PA metabolism on expression of genes in the polyamine pathway, that is from glutamate to ornithine to arginine pathway, and the entire transcriptome of the plant. So I'll cover at least with a few slides on some of the data that we have. To study the effects of altering polyamine metabolism on the entire metabolome and the transcriptome in the plants, to study the effects of increasing PA production on nitrogen and carbon uptake and biomass production, and to study the expression of polyamine biosynthetic genes in relation to abiotic and biotic stress and how they are regulated in plants. So what's our experimental design? Initial work was done with carrot and tobacco cells. Later on, we moved to the poplar cells, and we worked mostly with this clone of Populus nigra x maximowixii, clone NM6. This clone NM6 is of tremendous economic value because it is grown all over the world and millions and millions of hectares. And this clone is propagated only by cuttings. It does not produce any viable pollen grains, does not produce any viable seeds. So you can imagine over the last 40 years or so, since this clone of Populus by hybridization was made, it has widespread usage for biomass production. So that's the one we are trying to use. Initially, we started with cell cultures. Now we are working with whole plants. And also then to do some biochemical work nicely, we also worked with Arabidopsis. And we use constitutively expressed mouse ornithine decarboxylase gene or under inducible conditions. And I'll give you the idea of why we use the inducible and also constitutive expression. We have also produced transgenic plants with combinations of 2 or more genes. For example, both the ODC gene, which produces putrescine, and the SAMDC gene, which gives the decarboxylated SAM to produce spermidine synthase, and then the spermidine synthase gene. So we have plants that may be expressing all 3 genes to see if we can modify the pathway. Some are being expressed under inducible conditions and others under constitutive conditions. So we can control the pathway more precisely. And currently we are working on producing transgenic plants of the popular NM6 complete plants, which are, some of them are growing in our greenhouse right now, to study their carbon and nitrogen assimilation capabilities. So why are we using constitutive as well as inducible expression? Now, one problem which most people face it, however, they don't do much about it, with constitutive expression is that when you add a gene, the transgene is being expressed in every part of the plant uncontrolled. And in this case, let's say when we added ODC, the plants always had higher concentrations of putrescine. Now, eventually, we know that homeostasis, which is the regulated behavior of metabolism in the living cells, will make the plants adjust to this high level of putrescine, and we may not see the effects over several populations or several generations of a plant because the plants got so much used to high putrescine. So inducible expression allows us to precisely control the overproduction of putrescine at a short notice. And that way we can ask the question, what is the effect of short-term increase in polyamines? So in a situation like plant stress, polyamines go up in response to stress, and that is a transient increase. Now I compare these two promoters, which you may have not thought about it this way, that a constitutive expression of any gene is like having a product from the beginning of your life or being born in a rich family where there's a lot of money from the beginning. The day you're born, there's tons of money, and the day you're growing up and everything, you have lots of money. Inducible expression is you're born in a family which had low level of money, and then suddenly somebody in the family won some big lottery, and that lottery money lasted for a short time. You spent it, you enjoyed the life, and then you were back to normal. So inducible expression allows you to achieve a short-term increase and ask the question, what does a short-term increase in polyamines do to the cells versus a long-term stable increase? And I hope that if you have any questions about it, I'll explain to you more. So this is what the constructs we have prepared. Mouse ODC gene with an estradiol inducible promoter, selection marker being the kanamycin, or we use a constitutive promoter, which is the double 35S promoter from the virus with the same gene. Then we made a inducible estradiol inducible ODC gene and a constitutive SAMDC gene, or the reverse, that is constitutive ODC gene and inducible SAMDC gene, and all the combinations with the spermidine synthase gene or the spermidine synthase gene, and then produce hybrids which have all kinds of gene combinations. So you can see that we have plants that can almost do anything that we want with respect to the induction of a step in the polyamine pathway. This is what these the NM6 popular cells look like in culture. They are very funny looking, no relationship to the plant. However, now we have plants that are produced directly. These cells do not produce any plants. They are completely non-morphogenetic. Then we have the plants of Arabidopsis, which are producing those. These are the cell cultures of these popular plants. They grow very happily and very nicely. These are some of the technologies we have used and which I'll go through to give you an idea of where we stand with the results. So specific questions, which is kind of modification of the objectives of the study. What are the effects of mouse ODC expression on cellular polyamines and amino acids? Does the overaccumulation of putrescine alter cellular nitrogen-carbon ratio and total nitrogen and carbon uptake? Is ornithine limiting in the cells overexpressing the mouse ODC gene? Because as you will see, ornithine is not found in most plant cells in large quantities. So we are asking the question, would it be limiting? And we have the answer for that. How does increased putrescine accumulation affect expression of genes in the PA pathway? We talked about it. And how does increased production of putrescine affect other metabolites? So let's get into the results a little bit. So here's the initial question. What happens if you express the mouse ODC gene in plant cells under controlled conditions, that is controlled in this case, meaning, under uncontrolled conditions, that it is a constitutive promoter. So this is the life cycle of a 7-day culture. The amount of putrescine in these cells is in this range over 7-day life cycle of the cultures before their subculture. The high putrescine cells, which are expressing the gene constitutively, have anywhere from 4 to 5-fold or 6-fold increase in putrescine all the time during the whole 7-day cycle. Spermidine, on the other hand, may change a little bit, but not that much of a big change. Sometimes a little bit higher, sometimes a little bit lower, and spermine typically actually lower in concentration as compared to the control cells, which are non-transgenic cells. And similar results are seen with the Arabidopsis, except that Arabidopsis is a whole life cycle. The actual quantities could vary, but the ratios are very, very similar. Now, what happens if you use an inducible promoter? So here are the results of that. Here's the putrescine in a control, that is wild type plants. During this period of study, which is about 60 hours or so, we see no change, not much of a change in putrescine. But when you induce it with estradiol, you can see that you can get anywhere up to 5 or 6-fold or sometimes even 8-fold increase in putrescine with time. Spermidine, on the other hand, remains pretty much the same, whether they are control plants, non-transgenic plants, or transgenic plants. Spermine remains unchanged as well. But the mouse ODC, if you are familiar with it, also has a capability of converting lysine into cadaverine. This is what happens when the cells die. That's why you get this smell, cadaver smell from dead bodies. And that is because mouse ODC becomes very active, and since there's a fair amount of this substrate there, so you can have this cadaverine being produced in these transgenic plants, but no cadaverine seen at all in the non-transgenic plants. Here is the effect in the Arabidopsis on the total weight of the plant. Not a lot of difference. Fresh weight, dry weight differences. Dry weight also, it goes up a little bit in the transgenic plants in this line. The silique in the 2 transgenic lines is slightly significantly different, at least. The silique, number of siliques or fruits per plant is higher. The plant height is a little bit better, and the branching pattern is much more. So overall, there's an increase in biomass of Arabidopsis plants when you are expressing the mouse ODC gene in them. Then we ask the question, is the increased production of putrescine making it rapidly degrade also? And in this case, we use radioactive ornithine or radioactive spermidine in the culture medium or in the tissues, gave it to the tissues, and calculated the half-life of putrescine production or putrescine accumulation or spermidine accumulation. As you can see, if you feed the cells with the C14 ornithine, which will make putrescine in the cells, or feed them with C14 putrescine directly, the half-life of putrescine in Arabidopsis cells and also in the popular cell cultures is about 6 to 7 hours. And it doesn't matter which way you do it. If you have these high putrescine cells, the half-life is changed slightly. That's because of rapid production of putrescine. But basically, half-life does not change very much. And if you give putrescine to the cells, it's almost identical. Likewise, 2 things coming out of the spermidine study. Number 1, the half-life of spermidine is about 35 to 40 hours. That is, it takes that long for spermidine to disappear from the cells. However, in the high putrescine cells, again, there's a small difference in the half-life of spermidine, not a lot. Spermidine turnover remains pretty much constant. But it's interesting that typically spermidine and also spermine is even longer living, as about 50 to 60 hours. the half-life of these polyamines in the plants typically is a putrescine very high turnover, spermidine slower turnover, spermine even slower than spermidine. Then we looked at the activity of the enzyme that typically makes putrescine in these plant cells, and that happens to be ADC. Okay, so what happens to the level of ADC? Think of this, when we are making a lot of putrescine from ODC, one could expect that ADC activity is actually inhibited by high levels of putrescine. It turns out not to be the case. Actually, ADC is even higher. So when you're taking more putrescine away from the, more ornithine away from the cells, there is an increase in ADC activity. We can't explain it very well, but we know that there's a change in the mRNA of the ADC also, which means that the enzyme not only is being stabilized, but also being expressed more. So if you like, I can take a couple of minute break if you have some very quick questions. Otherwise, I'll continue. I know that typically we don't take a break, but if you would like to, I can answer some questions on the topic that we have covered so far.
No, go ahead, Professor. Then we can ask in the end.
Okay. So how are the total carbon and nitrogen accumulation affected? As if you recall, one of our hypotheses was if you increase the assimilation of nitrogen into polyamines, you might increase the assimilation of carbon along with that. And that is very clear from this experiment. Control cells, 6.82% nitrogen, and the high putrescine cells as high as 9% nitrogen accumulation as a percentage of their dry weight. And if you look at the carbon assimilation, that also, although there's a small change, but it is significantly increased. We have repeated this experiment more than once. It turns out to be there's always a small increase, which means the flowing in of nitrogen to the cells increases along with increasing the carbon assimilation. The only difference is these experiments were done with cell cultures, so the carbon was coming from sucrose. More recently, you will see the results of Arabidopsis, which is grown in the pots, and we find something very similar results. There is an increase in both nitrogen and carbon in the cells. And what happens to different amino acids that are part of the pathway that I talked about? Glutamate, which is the source of all ornithine, typically remains very high in the normal cells, and in the high putrescine cells, glutamate level goes down. That means a fairly significant amount of glutamate is being channeled towards putrescine production, and the polyamine increases. Likewise, if you look at ornithine, one would expect that ornithine being the substrate for ODC will be lower, and it turns out to be much lower. This is a 7-day culture cycle of the poplar cells. Arginine shows the same pattern. Arginine is also made from glutamate, so you can see that arginine also goes down. And proline, which is on the side of that pathway, if you remember it, it doesn't change very much. So the proline pathway is independent of what the effect is on glutamate, ornithine, and arginine. When you look at all the amino acids, this is the results of our metabolomic study and doing direct HPLC. We can find that different amino acids change differently in the control versus the HP plants on day 1 and day 5, those 2 days. The predominant amino acid in both cases is alanine, but alanine in the HP cells is almost twice as much, or 50 to 60% increase as compared to the control cells, and then at the expense of many other amino acids that go down. So, overall summary of the results so far. With the high putrescine production from using MODC, you are seeing increased demand for glutamate, increased protein accumulation, that is direct protein analysis. Mostly these amino acids will decrease and some of these amino acids increase. We don't understand the interaction of all of them with each other, but we know that some like glutamine, glutamate, arginine, that's much easier to explain than the other ones. So the question then shifted to how is the ornithine supply to MODC maintained? So if you recall the pathway, ornithine is the starting point for mouse ODC. It's taken away. What happens to the other enzymes in the pathway and their transcription, all that. So it was postulated that increased utilization of ornithine in the HP cells, which is high putrescine cells, would cause a considerable increase in its biosynthesis from glutamate and affect the metabolism of other cellular metabolites as well. The hypothesis was testing by transcripts of all genes involved in polyamine and ornithine arginine biosynthesis. The entire transcriptome was looked at and the entire metabolome, and that's where we have some results. This is the pathway we're talking about. We looked at the transcripts of all of these genes, and some on the sideline as well as the metabolites. We did not perform enzyme assays for many of these genes. However, we have used some glutamate C14 and tested that this pathway is enhanced. That's our unpublished data so far. When we look at the transcripts of expression, normalized expression of the transcripts of all of these genes, you can see most of them follow a similar pattern. And if anything, some of these, which are the red ones of these, are from the HP cells. Some of them are slightly reduced up front. This is again a 7-day cycle, 7-day study every day, this is a 1 day, 3 days, 5 days, and 7 days, every alternate day. You can see that the pattern seems very, very similar. So not much of a change in the expression of any of the genes of this pathway right here. Then we did compare using the microarrays at that time. This is earlier work. Various comparisons were made between control the HP cells and control in HP cells and the HP cells on day 3 and day 5. And we have now more data on Arabidopsis, which currently has one of the master's level student. He's writing his thesis. So data is still being analyzed finally, and I would not have access to all of that today. If you look at the expression of the different genes, you look at, this is using microarrays, not using the RNA-seq analysis yet. So you're looking at the express sequence tags, as they call them. There are about 448 genes out of about 10,000 or so, 9,557 genes. So many genes are increased in their expression, an equal number is decreases, and most of the gene expression, this is overall not related to any pathway, does not change. Likewise, on a day 5, very similar pattern, that maybe a little larger number. In this case, we have more genes here, so a little larger number of genes was increased. Not that much of a significance we can attach to this observation. These are again different ways of showing the same thing. Many of the genes that change, that are increased, are the same on day 3 and day 5. Likewise, many of the genes that decrease are the same, and others may be differently expressed between day 3 and day 5. These are the results of our metabolome analyses. The enzymes that they represent. A number of genes whose enzymes are changed are shown here. So the yellow ones are increased, fold, glutathione transferase, acetyl transferase, wound-induced proteins, a bunch of others are changed, and naming them doesn't matter. This is all published data. You can see it in some of our earlier publications. And another way of describing that you could actually differentiate between the HP cells and the control cells, green versus red, based upon a number of parameters on almost any day of the cultures. What it means is significant change in their metabolism and biochemistry has occurred just by adding this ornithine decarboxylase gene and modifying one step in the polyamine pathway. Some of the major metabolites that changed, GABA did change, ACC, which is involved in ethylene biosynthesis. You know, HP cells actually produce more ACC than the control cells. The alanine, as you saw, was a major change. Some other amino acids, GABA and everything else. These are the ones that are upregulated. And then there's a number of metabolites that are downregulated: asparagine, citrate, glutamic acid, glutamine, I already mentioned that, and so on. This is the overall effect of everything that we have seen so far. A summary slide. What does high putrescine do to the cells? Increase putrescine catabolism, increase ROS production, increase membrane damage. All these three things are possible, and membrane damage, the turnover of rates is much faster. The cells grow faster, but they are dying faster as well. What else does happen? Reduced potassium accumulation, enhanced calcium accumulation, increased nitrogen accumulation, enhanced carbon assimilation, reduced uptake of aluminum, increased efflux of aluminum, and enhanced calcium accumulation. All these are involved in protection against aluminum. All of these features are found in high putrescine cells. This is the change that we made. What else happens? Decreased glutamate. That is associated with decreased glutamine, histidine, serine, all these other amino acids, and increases in some amino acids. Of course, obviously, ornithine is utilized more rapidly, and we have seen enhanced ADC transcripts, enhanced ADC activity, and increased spermidine production in some cases, and catabolism, and so on. So this slide is kind of a summary of most of the things that we have seen. It gives you a good, this is a picture at the end of about 25 years of work. So, but coming back to the real practical advantages of this, here is what happens to Arabidopsis in different mouse ODC lines. You can see that the growth of the wild type plant is at a given time, and plants that are transgenic are slow in producing flowers, but eventually they produce tremendous amounts of flowers. And part of that reason may be that the flower production, once it is delayed, capability of the plant to produce flowers is increased. And these are the mature plants of these three transgenic lines and wild type. Whether the difference is bigger or not, but the biomass is bigger, as I showed you earlier. What are the morphological effects of overproduction of mouse ODC? In one line, at least, we found that there was a significant change in the pollen grain development. So if you look at the flowers and all that, the pollen development in these plants was much less, and the number of seeds produced in some cases was less, but that was not universally true. This is only one of the lines that we saw this thing. So what are the general conclusions based upon metabolic engineering of this type? Although the pathways for the biosynthesis of various amino acids are regulated independently, changing the biosynthesis or consumption of a single substrate like ornithine shows pleiotropic effects on the entire set of amino acid biosynthetic pathway. Not a surprise, because when you really affect the core metabolism, and ornithine is a part of the major amino acid, even though it's present in very small quantities, you can affect the whole metabolism of the plant. Since the primary target of genetic manipulation was increased utilization of ornithine and its accumulation in the cells is extremely low, it's obvious that in order to keep pace with its increased consumption, its biosynthesis is enhanced. You can imagine ornithine is very, very low, almost undetectable, and still you can see a significant threefold, fivefold, eightfold increase in putrescine. That tells you how quickly ornithine production must be happening from glutamate all the way. We postulate that cellular ornithine concentration plays an important role as a sensor or regulator in the signal transduction pathway that regulates its own biosynthesis from glutamate, which is the whole pathway that I mentioned, 5 or 6 enzymes. Furthermore, this regulation involves mostly biochemical steps and not increased transcription. So the machinery is there, the tracks are there, the train is moving faster. That's what our conclusion is. This pathway, acting as a sink of both nitrogen and carbon, results in overall increased nitrogen assimilation, which we have some evidence that I've shown you and some more that we are currently putting together. Objective 2, this is now we are next 5-7 minutes, we are changing our path here. We are interested in the regulation of expression of the SAMDC, 5 SAMDC genes and the 2 ADC genes using a slightly different approach, and that is the approach of promoter GUS fusion or a reporter fusion. Here are some constructs of SAMDC. Each gene, the promoter was taken along with or without the first few amino acid sequences or nucleotide sequences, and we asked the question, how do different constructs of SAMDC 1, 2, 3, 4, and 5 control the GUS expression? So very quickly, we studied the entire life cycle of the plant, and the SAMDC data is all published. You can see that. You can see the presence of the different SAMDC genes and the presence and absence of different parts of the promoter affect the gene regulation differently. He has very high expression of ADC, rather low expression of SAMDC4, and here SAMDC3, not much expressed in the roots, but expressed quite nicely in some of the parts. The end of the cycle, expression in all different parts of the flower express those 5 SAMDC genes differently. I'm not going to go into the details at this stage. This is something that I'm not going to go into. We have a number of other projects that a lot of undergraduates are working on. Production of taste-modifying cucumber and miraculin gene, production of new classes of antimicrobials, on and on. This is our training because our lab, as you heard, we have hundreds of undergraduates who have gone through our lab over the last 25 years or so, and they are working on very different projects from time to time. I've been teaching a number of courses, biotechnology and society, lab techniques in plant genetic engineering and biotechnology, lab techniques in physiology and biochemistry, cell culture, animals, which is an animal cell culture course, but I provide the aspects of plant cell culture. And this is where most of the students learn get their experiences from half a dozen or so undergraduate courses. These are the sources of funding that from time to time have been used to get the results that we have. Oops. This is our biomass that has contributed to our critical mass and collaborations from different places that have provided labor and brain to continue this work from time to time. These are some of the students who have recently graduated and are busy in their own work. And finally, thank you very much for your attention.
Thank you, Professor Minocha, for this nice talk. So I forgot to say a message in Portuguese to the students. I will just switch back to Portuguese just to let them know that they can ask in Portuguese and
Absolutely,
and I
yeah.
can switch back to English. Pessoal, se vocês quiserem fazer as perguntas em português, deixar no chat, fique à vontade que a gente faz a tradução para o professor Minocha, tá bom? So we can start with questions. I have to let you know, Professor, that the students, they normally need some time to warm up.
It's okay, no problem.
So I think I can start. I have to say that I do not have an extensive knowledge on polyamines, but I know, because I work on lignin metabolism, that there is this type of polyamine called thermospermine.
Yes.
And thermospermine apparently is involved in xylem differentiation. And I didn't see that you mentioned. I know that there's a problem to identify thermospermine because it's isomer of, I think
it's very difficult to actually separate it from putrescine.
But my question is, do you think that because you mentioned that one of the objectives of manipulating polyamines metabolism is to increase biomass, or to manipulate biomass? So my question is, do you think that thermospermine is a good target for metabolic engineering towards achieving higher biomass, for instance, in poplar, for instance?
Well, the question of biomass depends upon what your goals are. Biomass, if you are using it for timber production, then having a stem that is thicker and more xylem and more lignin is very useful biomass. If, on the other hand, you want to produce alcohol or energy, then you want more cellulosic biomass. Okay, so one would have to be very aware of that part. And most importantly, then you have to think about the regulation at the cell level. That's where the inducible expression or tissue-specific expression becomes very important. We didn't do tissue-specific, but we could regulate tissue-specific expression by adding estradiol to specific parts of the plant. Okay, and the third part is for xylem production, especially lignin production, the phenolics are very critical. Now, polyamines, which I did not go through because we have very actively growing cells and they don't make conjugates, but polyamines are very heavily conjugated with phenolics. Very commonly, and that phenolic conjugation in some cases is reversible easily. In other cases, as the cells get older, they just hold on to them. If you were to control the production of high amounts of polyphenolic material, which is used for lignin production, you may have to micromanipulate that you inhibit the conjugation of those phenolic compounds with the polyamines. So it gets more complicated. That's why there's future for research in any field. That's why we have the young people replacing us with time, continuing this work. It's not the end of the story.
It gets complicated indeed. Wow, I didn't know.
It does not answer your question specifically, but it tells you that there are multiple questions that are built into that question.
Oh yeah, good to know. We can try to find answers in the future. We have a question there at YouTube. I have to say that when I mentioned the message that Professor Eni Flo sent it to you, we were not online on YouTube, so I have to let Eni know that I forward your message, your hello, to Professor Minocha, Eni, and she has a question for you.
Sure.
She also asked about the importance of thermospermine, which I kind of already touched it, but she also mentioned or asked the importance of cadaverine for the plants.
Cadaverine in the plants is typically not produced much because the plant ADC, which is a major pathway for producing putrescine, does not utilize any other substrate. The animal ODC has itself a cadaverine synthase pathway. It's like the putrescine synthase or ornithine decarboxylase using lysine as the substrate. So if lysine was a major amino acid, then mouse ODC can handle this. In plants, cadaverine is typically not produced.
I'm sorry, she didn't mention plants. I said that, but
in animals, it is a very important thing. Now,
yeah.
here's a project that we somebody approached us to do. Maybe you can do that. That is, could you make your plants which produce cadaverine, and therefore, because cadaverine smells very bad, it may not be eaten by animals. Okay, because deer, for example, are very common for ornamental plants. They eat everything. Could you? I don't know if that's a logical way to think of it. That would you rather have cadaverine smelling or the flower smell that you like? But that tells you how complicated the things can be. But plants do not produce cadaverine much at all. Cadaverine can be produced from the decaying plants by bacteria, or animal cells produce it when they die. And this is one thing that happens in cadavers, the dead bodies of all animals. They produce tremendous amounts of cadaverine. And with regard to thermospermine, I should also mention, thermospermine is not detected in many plants, partly because we cannot detect it easily, partly because it's not high in amounts. In Arabidopsis, thermospermine seems to be a more common occurrence.
Also in poplar, you find it?
Poplar, we have not looked at thermospermine at all.
You know, because I mean, we had already some discussions here about how we use Arabidopsis as a model plant to everything else, but in some cases, Arabidopsis seems to be exception. So
it is. I mean, it is an exception. Arabidopsis does not have an ODC gene. Many plants have ODC and ADC genes. Arabidopsis has no ODC gene at all.
Wow.
So if those questions were addressed, we would not be using Arabidopsis as a model because its genetics is very, very unique.
Yeah, indeed. And he say that that you guys can make those plants together. I guess those plants producing cadaverine.
Well,
so you have a forest of cadaver plants.
Well, I mean, think of it this way. Maybe you can make cadaverine enough of it that the animals are distracted and you don't go near the plant too much until it flowers, and then cadaverine production stops. This is what the life of a designer of the plants is. You design a plant to do what you want it to do, not simply transfer a gene and say, well, what does it do? Like in my genetic engineering class, I asked the question to the students to write an essay on. And that question is, you see a plant, you wish it could, fill in the blanks, what you want it to do, and then you design that plant using the right kind of genes, right kind of promoters, tissue specific, induced, whatever, so that the plant can sing a song for you, or plant can do what you want it to do, not exactly what it does because you gave it a new gene and now it has no control over it.
Of course. Yeah, that's a great learning, of course. Professor, I have another question. I mean,
Sure.
I noticed that, again, I'm not an expert, but I noticed that arginine and citrulline, you mentioned, that are precursors to polyamines. But those, if I'm not wrong, they are also precursors of nitric oxide. So I was wondering, how is the relationship between polyamines and nitric oxide?
Okay. Polyamines, increased polyamine synthesis under certain conditions enhances actually the nitric oxide production, which has positive effects. Okay. On the other hand, under normal conditions, nitric oxide production should be decreased because you're using so much of arginine going through the pathway of making polyamines. Okay. So we have not looked at the per se nitrogen oxide production. We are just starting to do some work with using N15 labeled isotopes. And we might be able to get some idea of direct production versus nitrous oxide can be produced in there are several steps, several ways to produce nitric oxide. So I don't have the answer for that question at this stage.
Okay, perfect. Leandro, do you have any questions? I still have one more question.
Go ahead.
You kind of touched it when you were replying to me on my first question on thermospermine, but you showed the metabolomics data on your overexpressing ODC lines. And I was wondering whether you know what happens to the secondary metabolism in plants overproducing putrescine or overexpressing ODC. You already mentioned that polyamines can bind to phenolics, but what about like the overall effect on secondary metabolism?
Overall effect on secondary metabolism, we are looking into some, through the metabolome analysis. That's one direct way to do it. And the transcriptome, we just finished a study of the transcriptome of Arabidopsis control plants, inducible plants induced for 24 and 48 hours, and constitutively expressing plants. So the student is looking at some of the enzymes in the KEGG pathways, but we have not finished the analysis yet. So cannot tell you exactly what happens in our plants. However, other people have done some studies and they find that secondary metabolites, especially the phenolic pathway, is modified significantly.
Yeah, I guess also because it modifies the levels of amino acids, and phenylpropanoids are produced from amino acids.
Yes, so we know the amino. When we do our polyamine analysis, we have a system where we can do polyamines and all amino acids in the same HPLC run. Okay,
cool.
And that is something that tells us we can see about 16 amino acids, 16 or 17, because 2 or 3 overlap. Their separation is not easy in the system that we have. But we just then put them together and say, this combination of these 3, some of them are in very small quantities, others are significantly large quantities.
Okay, Leandro?
Yeah, well, I have a lot of questions, but
it's okay.
I will focus on a few of questions. It's very interesting about the high levels of alanine.
Yeah.
And what do you think about the relationship between alanine and polyamine metabolism?
I've not thought about it. It's just an observation. So at this stage, I won't make a hypothesis. I would, again, when you look at the relative ratios of different amino acids, some of that could be related to the ability to look at some of the amino acids that are low quantities from the metabolome analyses. We did not do our own metabolome analysis to see how accurately they were measuring all the amino acids. But in our own system, when we do the HPLC analyses, we know that some of them are not easy to separate out. But why does alanine show up? It's one of the smaller amino acids. I don't know. I don't know what's the regulatory aspect of why, relatively speaking, absolute quantities of alanine are higher, not simply as a ratio of the other amino acids.
Okay. And another question is about it's very interesting, the result about the increase the putrescine and increase the carbon assimilation is very interesting for application in crops.
Yeah.
By gain of biomass. So, but this is a particular case in Arabidopsis or poplar, or is a general effect of the putrescine?
If you look at direct effect, that is not including stress tolerance or anything of that type, there's not a lot of data on that.
Great.
Okay. And especially when you're looking at tissue specific, that's what we are going to do next, the leaves versus stem versus roots and all that. So at this stage, the only thing we have done is with the Arabidopsis and the cell cultures of poplar, the plants of poplar which have these genes have not been tested yet, or we are in the process. We just have them growing in the greenhouse. This will be the first summer that we will get a good idea of the biomass of those plants.
Great, this is very interesting. Well, it's a particular question is about the two enzymes, ADC and SNDC, because in most of studies papers, these two enzymes is the main enzymes of that regulate the biosynthesis of polyamines, right? Because the spermidine and spermidine synthase not even is the most gene activate or high expressed. So do you think that these are the two enzymes that control the entire metabolic pathway of polyamines?
Well, the spermidine and spermidine synthase do require fairly high activity of SAM-DC, the availability of SAM, acetylmethionine. Then we looked at, this was not our own work, but in collaboration with some people with the USDA, the question of ethylene production, which also is produced from acetylmethionine. So we were looking at the competition between, if you make more polyamines, would you reduce the ethylene production by tomato fruits? Okay. Yeah. And we found that there was not much of an effect. There was no direct competition. There was enough acetylmethionine being produced by SAM synthase that it could handle the maturing fruits for the production of ethylene while still maintaining polyamines. If you inhibit polyamine biosynthesis by reverse methods, genetic methods, still the ethylene production could be controlled only by directly inhibiting ethylene biosynthesis, not by indirectly stealing away SAM.
Okay. My question is because it's very interesting to express ODC in high levels of putrescine, but I think that if it is interesting to attack in the SNDC. So is it is dependent or independent of the upstream, the pathway, or not? Because the SNDC is part of that compete with ethylene metabolism.
Yeah.
It's a little bit polemic, this part, I know. But if it is, what's the importance of the SNDC in like a response to stress or to gain in biomass on a crop?
Well, I would think that since ethylene is important at certain steps of growth of the plant or development of the plant, for example, fruit ripening is one, senescence is another one, germination of the seed, ethylene plays a role in opening up the curvature of the hypocotyl and so on. I would think that ethylene is controlled a bit more independently of the polyamine pathway, because some ethylene production is enhanced at the time when growth is overall inhibited. That means the polyamine levels are going to be low. The regulatory aspect of that being growth being regulated, not polyamines being regulated to slow down the growth. Okay. That's my hypothesis at this stage, because we know that when we look at the older leaves and all that, polyamine biosynthesis, because of the transgene, at least continues. Mm-hmm. And the plant is still senescing.
Okay. And I have two more questions.
Go ahead. I'm okay.
Go ahead.
Too early to go to bed.
In the recent years, is a lot of papers about the GABA. Is the popular amino acids now?
It's a very important metabolite. Yes.
Yeah, and my question is, what's the next step about the polyamines metabolic pathway? Because we know very well about the biosynthesis now, after your works, after other works and but I think that we don't know enough about the catabolism and the effect of a catabolism of polyamines in under very conditions of growth or cell differentiation. And I think that about the catabolism, like peroxidase is a product of catabolism of polyamines, but it's very interesting about the GABA, because now we have more studies about the main role of the GABA under stress. And what are the next steps?
It is. I agree with you. I think the question is very important. The two pathways, one is very direct, glutamate decarboxylase, and the other is a very indirect pathway. Reading all the most recent papers on the topic, I gather that direct production of GABA is very important under normal conditions. One step, one enzyme, regulated as much the demand there is. However, when the plant is in response to stress and all that, where it's affecting the polyamines, knowing that the half-life of putrescine is rather short, there's a fair amount of GABA production from the polyamine pathway, which also is using glutamate. So the question is, does the GAD pathway for GABA production decrease because the cells want to regulate GABA production as a combination? That question is still, in my view, not answered. Okay. So I think one of the students is hopefully going to do some experiments with these transgenic plants under inducible conditions, because that's where you can answer the question much better by using the dual isotope labeling of glutamate or urea. So I think that's the only way to follow glutamate, by labeling it at different places. So we know what part of glutamate is going through putrescine and what part of glutamate is going directly into GABA, and then looking at that part of it.
It's a I don't know the word. Well, it's okay.
But I think there are two hypotheses to look at. One is the cells control the GABA production. They don't care where it comes from.
Okay, it's a compensation.
Okay. The other is that the GAD controls GABA production and may not have a indirect control over it, because increased GABA does not affect GAD activity. So far, it looks like GABA production from the polyamine pathway does not really affect GAD activity. But then it doesn't affect the expression of many other genes. So the activity may be related to the availability of the substrate. If the substrate goes more on one side, the other side just slows down. As long as there's a significant amount of glutamate, then both pathways work. So we're looking at that part right now. Hopefully we'll have some indication of what's going on, but at this stage I don't have any, I haven't not seen a publication directly on following the glutamate molecule clearly from both pathways.
Right. My final question is a philosophical question. What's the next steps, Professor? In your career, in your research?
Well, in my career, I don't know. I have another couple of years before the students finish up. By that time, hopefully, travels will increase. So we'll do a bit more travel. And one day at a time. Rakesh is still working for another year or so. So by that time, she'll be retiring too, and we'll see what we can do. We'll come over to Brazil and spend spend our winters there.
Exactly. drinking caipirinhas at the beach.
There you go. See?
Okay,
but
so
some of you can come visit us, as long as we'll have the lab and all that, or otherwise also if you come to US, let us know. We're planning to stay in this area. We are not moving permanently to any other place. So you have a place to stay.
Thank you. So do we have more questions, guys? I think on YouTube, we do not have more questions. Here, we still have some times for questions if the students have. If not, I don't know. Nobody is sending anything. So Well, I think it's enough. So I would like to thank you, Professor Minocha. It was super nice to have you here. It was a very interesting talk and a very nice conversation. So I would like to thank you once again in the name of the whole team, me, Magda, and Leandro. And yeah, if we have the opportunity to make it real, what you invited us, or if you come to Brazil, you can always visit us at USP. So
Absolutely.
thank you very much, and
Yeah.
I hope you safe and healthy.
Thank you very much. And
Thank you,
be
Professor.
positive and stay negative.
Exactly. Thank you very much. Thanks everyone for the attendance.
Yeah, thank you very much
Bye-bye.
for attending it. It was wonderful. I had a great experience. If it was live, it would have been even more fun.
Exactly. Okay, so thank you.
Yep.
Bye to everyone.
Bye.
Okay, bye-bye.
See you the next week.